This is my third National Dahlia show, so I guess I can't feign ignorance anymore. I've become a Dahlia person now. I never thought I would say that, and I've asked a bunch of you, what got you into Dahlia? What was the spark? What was the one thing? And you all tell me a different story, but it all revolves around this same idea of you grew a flower, you talked to somebody, you met a breeder, you met a grower, and it clicked, and you fell in love with the diversity. That's not my origin story at all, actually. I was tricked into studying Dahlia by a professor, a very brilliant professor at Stanford named Dr. Virginia Walbett. She asked me when I was a postdoc, could I help with the Dahlia Genome project? I said no. It sounds really complicated. It's an octoploid, it's a big genome, I have other things to do. She said, great, I'm gonna send you a box of tissue. I'm retiring probably pretty soon. So this is now your project. And sure enough, 3 days later, I get a box in the mail from FedEx on dry ice with seeds, tissue, diverse collections of dahlias from several species. And I didn't realize it at the time, but that was a turning point in my career in many ways. My laboratory studies how plants reproduce, how plants have evolved and diversified over time. And in many ways, Dahlia has become an incredible story about how plants can evolve and diversify in such brilliant ways, and how we can use genetics and genomics to tell those kinds of stories. So today I'm going to give you an update on the Dahlia Genome Project, and I brought with me a PhD student in my laboratory who's funded by the American Dahlia Society, Zach Maharg. We're going to tag team this talk together. So I'm going to give the first half, and then I'm going to pass it off to him. So I'm on the faculty at a biotechnology institute in Huntsville, Alabama. You might know Huntsville for Space Camp. They have adult space camp, by the way, if you feel like you missed out as a kid. You can go there, they have an open bar and stuff like that. You can sit in the rocket and have a beer and literally drive to the moon. But there's also an incredible biotechnology arena in Huntsville, Alabama, one of which is Hudson Alpha. On campus, we have the world's longest DNA double helix. Hudson Alpha is renowned because, does anyone here remember the Human Genome Project?
Oh, yeah.
Yeah, a lot of people. Thought of in the mid-'80s, took nearly 20 years, $3 billion, tens of thousands of human geneticists around the world. It was like the greatest international collaboration. We all came together, multiple countries, to sequence one human genome that was going to change the world, allow us to find the genes that control major diseases in human biology, and it turns out it was far more complicated than that. After the human genome came out in 2001, and then again in 2003, many of the scientists who worked on that original Human Genome Project picked up shop, left Stanford, and founded Hudson Alpha Institute for Biotechnology. So this place drips in history, going all the way back to when these first sequencing and genome assembly technologies started to emerge. And if you come to Hudson Alpha, this is a global leader in the genome sequencing of complex plants, animals, and humans. We have rooms filled with these genome sequencers. They each cost $1 million or so. They're constantly running. I think we've got 30 in the building. We can sequence 1,000 human genomes in 2 days, or 1,000 corn genomes in 2 days. So imagine the capacity now. We can speed read DNA of organisms to look inside of cells, like Dr. Papu was saying, and understand the genetic code of organisms. This is fundamentally changing the way that we look at biology. We don't just look at plants from the outside anymore. We look at their DNA at the exact same time. And we start to make connections between those two. A show of hands, who has done a 23andMe or an Ancestry.com or anything like that? Probably about a third, half the room. Yeah, something like that. It's kind of an amazing business model. You spend $100, you buy the kit, you swab your saliva, you send it back to them. They send you a report that looks something kind of like this. Your ancestry composition. Sometimes you find out your parents aren't really your parents. Sometimes you find out you've got a long-lost brother. I mean, it's amazing what our DNA has done to change the world. If you're the Golden State Killer, now you get caught by your DNA. It's really amazing, actually. And it's something unifying everybody in this room, everybody on our planet. Plants, animals, fungi, all of you. We share this incredible common connection of our DNA. Anyone have dogs? You can do the embark. Same thing. And you get back this amazing report that shows you what percentage different kinds of breeds your dog might be. Is it a purebred? Is it a mix? Is it a little bit of everything? Be nice. This is Zach Maharg's dog's report. If you're a cat person, I don't want to bias anybody in here, you can also do wisdom panels for cats too, and figure out that Violet might be 5 breeds, mostly American domestic cat, maybe a little Norwegian, and 2% Munchkin. I'm not sure what that is, but it sounds adorable. These companies all do the exact same thing. They will take your DNA from yourself or from your pet. They will generate a little bit of genomic data on it. They will put it into a large database. They will mine that database, give you a report, and then they keep all the data. And that is an incredibly powerful database that they have access to because they have DNA, they have genes, they have mutations, and they have phenotypes that go along with it. The ways that something looks. Whether your cat is prone to scratching, whether your cat has allergies, whether your dog hides behind couches, all of these kinds of things now, you can connect genotype, meaning your DNA sequence, to the phenotype, how that organism actually looks. And the key question they all ask, does a genetic variant correlate with a certain trait? What if we could do this with Dahlia? What if we could predict what a Dahlia cross would yield? What if I could tell you, if you give me a leaf of a Dahlia, I could tell you its ancestry. I could tell you if it had certain patterns of potential disease resistance. I could tell you eventually what its flower would look like in terms of size and form and color. Does it have certain markers that correlate with cut flower longevity in a vase? What if we could make a report for something like an NSE, where you send me a leaf and I tell you exactly what the ancestry, what the history of that plant is throughout its long history of hybridization for Dahlia breeders? To do this, you need three things. And what I want to convince you of today is that this is a very doable thing using genetics and genomics. We can create a 23andMe type environment for Dahlia. We can increase your ability as breeders to make thoughtful crosses that produce interesting offspring that you want to breed for. This is how, in all major crop species, corn, cotton, soybean, peanut, this is what we do. And so this is not a novel idea by any stretch of the imagination, but there's a few things that you all don't have in the Dahlia community that you do need to be able to do this. The first is a reference genome, meaning we need to, just like the Human Genome Project, put together all chromosomes of a genome in order. The second thing you need are phenotypes, the ways that plants look. You need diversity, in other words. And the third thing you need is for those same individuals that you phenotyped, you need genetic data, meaning you have to understand what kinds of mutations are found in those certain organisms. And then we can find these gene mutations that correlate with certain phenotypes. We can build genetic markers for them, and we can screen large amounts of germplasm to say, this individual has these three markers that I think typically associate with this kind of floral form. This is, in my opinion, part of the future of horticultural breeding. This first step, how do we assemble a reference genome? I hate puzzles, actually, and it's a very confusing career that I've got myself into because all my laboratory does is assemble puzzles. We just call them genomes. And the way that you generate a genome nowadays is you make puzzle pieces. We extract DNA out of an organism, and then we put it on a DNA sequencer. You build these puzzle pieces, and then you teach a computer how to put them together into chromosomes. That took the Human Genome Project 20 years and $3 billion. It takes us, about a graduate student's, a couple months of time and maybe $10,000 now for small, non-complex plant genomes. It's incredible how the field has changed in the last 25 years. And what do I mean when I say a puzzle piece? This is an actual piece of data that came off of a sequencer in my laboratory. It is just 4 letters, A, T, C, and G, the 4 nucleotides that make up DNA, the building block of life. We are not all that complex, it turns out. In every single cell of your body, you've got 23 pairs of chromosomes that have sequence that looks just like this. And so I hope that kind of anchors you that we are not more or less advanced than any other organism on the planet. We are, by and large, 4 nucleotides put in different orders over and over and over again. So if we're going to do this for a Dahlia, which Dahlia accession of a particular cultivar, I had to get this right, I think, do we pick? What actual plant do we want to sequence? We had a bunch of ideas. This genome project has evolved a lot in the last 5 years of what was feasible, what was possible, what actual plant do we want to generate this high quality genome for. And so with the help of Christine and Brad and a handful of other folks from several of the societies in the country, we wanted to see how complex a handful of different Dahlia species are. There's maybe, 43 species in the genus, give or take a little bit. What is a species? Kind of confusing question to begin with. So we generated DNA sequence data for about 6 individuals. Dahlia rudis, Coccinia, brevis, sorensenii, and 2 of the variabilis species, Thomas Edison, which is an older cultivar, and Edna C. And we can generate just a little bit of DNA sequencing data that allows us to build these plots, like you see on the right side, that tell us how complex a genome might be. Is there 1 copy of every gene in a genome? Are there 8 copies of a gene in every genome? Is it diploid, tetraploid, octoploid, and so on? How big is the genome? How much is it going to cost for us to generate a genome for, etc.? How much of Zach's life and stress is going to go into putting together a genome like this? When we look at, for instance, Thomas Edison, we know it's an octoploid from this data. There's 8 copies of every single gene in the genome. In contrast to humans, like Dr. Papu said, where we get 1 copy of every chromosome from mom, 1 from dad, Dahlias get 4 from mom and 4 from dad. So it's incredibly interesting, fascinating, and very difficult, actually, to do a genome project for a species like this. But this doesn't scare us. And nowadays, the sequencing technologies at places like Hudson Alpha, where we are so good, where we decided to go for it. Let's pick the most complex and useful and interesting thing that we can pick to build this idea of a 23andMe for the Dahlia community. Our takeaway is, yeah, they're all going to be complex. Just pick the one you love the most. And so we asked around. And now we're generating a genome for Edna C, the Dahlia of the half century, as it's been called. Iconic, of course, in many ways. But again, we're not sequencing the wild species. We're not sequencing a tetraploid from Mexico or anything like that. We're sequencing an octoploid genome. Same species as pretty much every flower at the show. So in order for us to generate a genome, we have to grow up plants. Christine Albrecht was kind enough to send us many individuals of clones of Edna C. Hudson Alpha is not just a genetics and genomics institute. We do get dirty. We just built a new 13,000 square foot glasshouse complex that's got molecular lab space in it, and all these beautiful glasshouse rooms that have full supplemental lighting 365 days out of the year. So we have broad spectrum LED arrays, far red, temperature and relative humidity controlled rooms. So we can grow dahlias year round, in other words. And actually we can grow a whole lot of dahlias year round. As you can see, one of our largest rooms is actually filled with Zach's dahlia plants right now. And again, this is the product of his PhD work. This is one of my favorite things to show visitors to the Institute of the impact of what we can do with genetics and genomics. So now I think we've got 50 accessions across 8 species. I was pretty close. Maybe it's pretty good. So we have a lot of dahlias growing in our current greenhouse, which has been really exciting. So we're seeing all this floral form diversity. We're growing up multiple species. As Zach is going to tell you in his part of the talk, we're building phylogenetic trees to understand how dahlias have evolved. Here's another photo of Zach, looking really cool. So the first thing you got to do is you got to take leaves of that Edna C, and you have to grind it up. Plant cells have a cell wall that you got to crack open to get DNA out of it. So we use liquid nitrogen, we grind things up with mortars and pestles. And this is what DNA looks like. It's rather unsatisfying, actually. It's a tube of clear, mostly colorless liquid that holds this entire book that we're talking about. And the trick for generating really good plant genomes is that you need to extract really big pieces of DNA. Has anyone done a strawberry or banana DNA extraction in a Ziploc bag before? No? None of you? Yeah, a few of you, maybe like at summer camps and stuff like that. We do the exact same thing. And I know that might be kind of weird to think about. We use a little bit of detergent, a little bit of soap, and we grind stuff up, and we smash up the cells, and we get big pieces of DNA out. And you gotta be really gentle with that DNA. It's almost like an art form, actually. And when we get huge pieces of DNA out of an extraction like that, we get big puzzle pieces. Meaning I can take that DNA that we extract, and we put it on one of our fancy genome sequencers at Hudson Alpha. The same machines we use to generate perfect human genomes, we're using to generate perfect plant genomes now. So in many cases, we're getting whole chromosomes in 1 or 2 pieces now. It's really an amazing time to be a plant geneticist, because now we can put together the entire length of every single chromosome in a plant genome that we've never even seen before, something that we've never done any research on in just a matter of a month or so. So right now, Edna C is currently on one of these sequencers. Here's a photo of Ida loading the sequencer with Edna C on it. So our estimated timeline is that in about a month, we're gonna have all the data generated we need for this Dahlia Genome project. The time has finally come. I've given 2 national show talks where I've said, we're close, but now we're like there, right? I promise. And in February of 2023, I ballpark that Zach will probably have the first version of our genome assembly done by then. An incredibly exciting time for the Dahlia community. That's step one. Second thing we need are phenotypes. When I walk around this show, I look in every single room and I see an incredible amount of diversity. You all have already done all of the hard work. And I mean that in the kindest way possible. You have already built a phenotyping catalog for every single bloom, and it's in those books, where you totally nail out the size, the form, what kinds of colors these plants are. These are the kinds of things that take plant breeders years, decades sometimes to do. Hanu talked, for instance, about it's hard to phenotype a plant for resistance to virus. Well, it's easy for us now to look at this kind of sheet and look at all the phenotypes for color, form, and size. Again, that's because of the community that you all have built. So last year, or 2 years ago? Last year, oh my gosh, time. Last year, you might remember us taking photos and leaf samples from every single plant at the Dahlia show. Does anyone remember helping with that? There's a few people, yeah, a couple in the room were helpful there. So we walked around with silica packets and we collected a photograph and a leaf from every single Dahlia at the show. Wow. So imagine this. You all have done all the phenotyping already. You've told me the color, the size, the shape, the variety name that you've picked. And then now I have access to leaf tissue and a photograph for every single one of those individuals. Does it sound like 23andMe yet? A little bit, right? And what Zach did is he took all these photos that he collected at the show, nearly 1,000 in total, and look at the diversity that you all bring in one place. Talk about a gold mine. You walk into one place, one hotel, and you get this diversity, and we can collect DNA and leaf tissue from every single one of them. Imagine you're a plant breeder. You do that in the field over generations, over tens of years. And what took breeders that long takes us about an afternoon with help from you all. So now we've totally smashed apart the 23andMe model, actually, because you all have done all the hard work. Back at the laboratory, Zach organized all of these packets that you all helped us collect. These are in our laboratory. We're extracting DNA from them now. The third thing you need is that DNA information. You need genotypes. So what we do is we take those leaf samples, we extract DNA from them, we generate what are called short reads of DNA sequence, very cheap, inexpensive ways to generate genome sequencing data. And then when we have our reference genome, for every single one of those 1,000 individuals, we're going to align that DNA sequencing data back to that reference and identify mutations. For instance, in the reference genome here, we have an A nucleotide, and in all of the short reads that we generated for a given individual, we have T's, meaning at that position in the genome, there's an A to T mutation. That mutation could do something, could be important for something. And the way we ask that question is through statistics. We ask, is there a statistical association between a given trait, like floral size, bloom color, whatever it might be, and a mutation that we find in that sequencing data? This is called a QTL, or a quantitative trait locus. And when we plot for every single gene in the genome, for every single mutation that we find, are there certain mutations that are always found in some traits? Meaning a mutation that always is found in every red bloom, or every large bloom, or every microbe, or whatever it might be, every cactus, and so on. And this is the way that the plant community finds genes that control major traits. And in this case, it could be flower color, shape, longevity, disease resistance, drought tolerance, and more. This is how we find genes that control major traits in any plant species. But in particular, this is what we're doing for Dahlia. So why does this genome matter? And I want to pose kind of a provocative question. When I look at your society, when I walk around this show, for instance, I walk into every room, I see all the amazing diversity that you all have put your souls into. And I think as a geneticist that is an incredibly valuable thing that you all have done. And what we can do as geneticists is extract value from the diversity that you have put into one room. So imagine if we could generate genotypes for all of these individuals, which we're doing. We develop genetic markers that are linked to major traits like color, size, bloom shape, whatever it might be. And then those are markers that can be licensed to seed companies, like Ball, for instance. And perhaps ADS could share in the royalties of that kind of model. Because again, you all have done the hard work. And then we as geneticists are helping to extract value from those kinds of collections. So I think it's an interesting idea that I want you all to put in the back of your minds and consider that this is not just a hobby. What you've done is created an incredible amount of genetic value, of statistical power, of the ability for us to find genes that control traits, not just in Dahlia, but actually in many Asteraceae species. The quote that I want to leave you all with before I pass this on to Zach is that one of my favorite things to say is that within each of us there is a book. And that could be describing your experience growing up, your shared experience as a human being. But within each of us, you have a DNA sequence, a story that tells quite a bit about you. Plants have been around since the first green thing evolved about 1 billion years ago. Plants have seen quite a bit on our planet. Meteors hitting the planet, wiping out all animals except for the lineage leading to birds today in many cases. Plants are incredible survivors. And what we do as geneticists is we use DNA sequencing to tell those stories. And so that's where I want to pass this on to Zach. Zach is a PhD student funded in my laboratory by the American Dahlia Society. And he's built an incredible set of dissertation chapters, from assembling the genome of Dahlia, identifying these mutations that control major traits, and what he'll talk about today is building phylogenetic relationships, of understanding how Dahlia species are related to each other. I met Zach at the first national show in Grand Rapids that I went to. He came up to me after a talk I gave and didn't even introduce himself and said, "I don't know what I want to do. I just graduated with a math and bio double major, but I think I want to do that." And so I owe a lot to you all for helping me find such excellent students like Zach. So I'm going to pass it off to Zach now. Thank you.
So as Dr. Harkess said, this is my second year of my PhD at Auburn, and today I'm going to be talking about how I'm creating a phylogeny to try and parse out what Dahlia species had to have hybridized to create the Dahlias that all of you brought today. So on the screen right now is a phylogeny, which is an easy way to think about it is like looking at your family tree, but just all of like your cousins are labeled at the end. So each of these little tree splits would be like your grandparents or your mom, and all of the labeled would be like your siblings and cousins. So in the Dahlia genus, there are roughly 42 species, ranging from tetraploids to octoploids. So that's what on this graph, anything that says 4X would be a tetraploid, meaning it has 4 copies of each chromosome. Anything with an 8X means it has 8 copies of each chromosome. And for the most part, all of the octoploids tend to be in this part of the family tree, with a couple exceptions of some species having tetraploids and octoploids of each individual. So what I'm trying to understand is what had to have happened within the genus evolutionary time-wise to create such this wide diversity of tetraploids and octoploids. So all the green little dots or circles on this phylogeny are areas where I think something had to have happened within the genus to create a change in ploidy level or a change in chromosome numbers. As you can see, for the most part, these all have 32 chromosomes, but then you have a couple down here that have 34, and you have this one species that has 36. That means somewhere over here between these two species, something had to happen to create a Dahlia with 36 chromosomes instead of 32. On top of that, so one of the main hypotheses of the parents of the cultivated Dahlia is that it came from 2 different groups. Group 1 Dahlias are these are all species of Dahlias that make purple, white, and lavender flowers. And all these red ones are ones that make the red, yellows, and oranges. And the only species on this tree that creates both is Varibalis, which is the cultivated Dahlia that all of you know. So it is thought to believe that something from group 2 had to have hybridized with something from group 1 to create this cultivated Dahlia. And to find that, I'm looking at reconstructing this phylogeny with sequencing technology at Hudson Alpha, as well as getting a hold of all these species and trying to make these crosses in the greenhouse, or if things go well with all of your help out in your gardens. So I thought it would be important to show the different species and the flowers they make and where they are in the phylogeny. So it is thought to believe that Coccinea, which is this L, M, and N, these ones, is the parents to the group 2 Dahlia. But the papers I've been reading, no one is quite sure what species from group 1 was used to make the hybridized cross. And I'm starting to think that just by looking at all your dahlias out there, that there could have been multiple hybridization events that have occurred to create such that wide floral diversity. So to do this, I'm going to use a technology called target sequence or bait capture. And what it does is it looks at certain genes that are found throughout a common family. So I'm using one that is specific to the Asteraceae or the sunflower family. And it's going to look at genes that are only specific to that. So it's not going to look for like anything else other than like having the type of florets that a dahlia has, or having the way the blooms progress. So then what it does is you put these little beads into our DNA extractions, and it'll find the genes that we want to target, bind to those, and then we take a magnet on the side of the tube, and the beads pull away the DNA that I want for each tube. And then we do we put it in some like ethanol to just wash all the beads off, and then we put it in a thing called PCR, which will just replicate the amount of DNA over and over again, and then we'll sequence that. And what that'll do is I can do some computer analyses, and it'll tell me this species is this closely related to this one. And this is relatively cheap, as it's about $20 to do one sample, which is drastically cheaper than doing a full genome assembly for every single dahlia out there. And so this is basically what it would look like once I get all my data back. I'd have gene 1, gene 2, and all the way up to all my genes that I have. It looks at all the nucleotide differences between all these genes. These are called gene trees. And then what the programs do at the end is they put all of those gene trees together to create a pretty accurate phylogeny with different splits based on how genetically related all these species are. So to do this, I need to get access to all the species out there. And they're not relatively easy to find. Like, you can't just go to the local hardware store or garden center and just buy one of these species. So what I've been doing is working with herbariums to get sheets of dahlia leaves just like this. And what we can do is take just one of these leaves about the size of half my thumbnail, and we can extract DNA from that and get enough DNA out of it to put into this bait capture technology. This is just a short list of all of the herbariums that I've been reaching out to and getting species for. And at the moment, I have about 37 of the 42 species from about 6 or 7 different herbariums around the country. I think the coolest part about doing this project is I actually got in contact with Dr. Sorensen, the one who helped helped revise the Dahlia genus, and I got a lot of herbarium supplements from him, including a couple of isotypes, which are basically just an herbarium sheet that is used to name a species. So then most of the time when you do these kinds of projects, you really don't get those. So I was really honored that he trusted me enough to send me those. So currently in the lab right now, we extracted DNA, and we're going on to that. We're going on to this step of the project. So I'm hoping within the next few months, I should be able to have a full phylogeny. Wow. So that leads up to our summary of the genome is underway. It's being sequenced right now as we speak. We have over 1,000 dahlia photos from this show and the last show, along with phenotypes for about 537 that were at the show last year. And we're going to be starting DNA extractions on all of those shortly. And that's about it.
Wow. Oh, you missed the good point. Zach is a second-year PhD student now. So this has been a pretty amazing project. My laboratory is a fledgling lab. I started March of 2020 at Hudson Alpha, and so we work on a diversity of projects, and Dahlia has become something that my lab enjoys deeply, actually. We've obviously got a dedicated person working on it, and we benefited greatly from all of your enthusiasm germplasm, material, advice, and certainly funding from the American Dahlia Society to have a person to actually work on this in a dedicated way in our laboratory. So we owe really most of the credit for this project to the Dahlia Genome team. And so Christine Albrecht and Dr. Ginny Walbott, I think, were the two main instigators of this back in the day. And this is probably a 10-year-old, maybe longer idea that now, with new advances in sequencing technology, is more possible than ever. And so when I'm glad that this project didn't generate a genome until now, because we've got the tools to do it better than ever. And so I'm grateful that, yeah.
When you go for samples, there's section S, which is singles, and then the ABS is single bench. These are new hybrids that haven't been introduced. So
Cool.
be sure you focus on the seedlings to get information you won't get anywhere else.
Did you bring seed packets?
I did not this time.
Okay, yeah, we'll get some. That's awesome, thank you. The power for us as geneticists is that you all have diversity here, right? When you have diversity of phenotypes, you likely have diversity of genotypes. And when we pair those two things together with an ultra high quality reference genome, that's how we find genes that control traits. And again, leading back to this idea, once you find a gene that controls a trait, you can do something with it. That's valuable. You can use it in a selection. You could run a marker on all of your individuals in your breeding population and ask which individuals have that marker that I want, have that gene mutation that I want. Then if I cross them with another individual that has a different kind of beneficial trait, that's how you accelerate plant breeding programs. Again, this is what they do in all the major crop species. So I think this society is sitting on a gold mine that horticultural seed companies would kill for. And in fact, I know they would.
Should we worry?
No, I think you should be excited because I think there's money to be made in a new way that could sustain horticultural societies in a different perspective. Yeah.
Yeah, so to that point, who owns the data?
Who owns the data? So in this case, you all did. So that's a conversation that we definitely, who's the lawyer?
We didn't make this many money.
Yeah, that's a fantastic point. And for right now, we're just trying to map, major QTL for some of these traits, but certainly there's a chance we could find genes that are valuable. Again, it's only valuable if somebody wants it, I guess is the other question, and it could just be beneficial to you all as breeders.
I think, again, at this show, and I saw last week at the show in Philadelphia, in Baldwin, there's a Dahlia from 1940 that looks like it has perfect foliage. It seems like to not be affected significantly by the virus issues that we're all dealing with. This resistance thing is really a big deal too, so we don't get so frustrated with stock. But I think a project involving the phenotype or whatever, and the genotype that goes with it, of this resistance thing that we all have. And the people they're amazed at some of these older dahlias, 20, 30, 50 years old, that still look as good as they did then.
Yep, Little Bees Wings, Thomas Edison.
We need to work with you, especially on that resistance thing.
Yeah, I'm certainly not a pathologist, so luckily, Dr. Papu is, of course. And so I think there's huge potential to leverage old accessions like what you're talking about, herbarium sheets, diverse species from Mexico as well, and start doing this kind of testing. Again, when we look at the larger diversity of the genus, do we find resistance outside of just this one species that mostly you all grow?
I thought years ago, one of the questions was how much, if any, do the dahlias today differ from the ones that are growing in Mexico hundreds of years ago? What is the simple answer to that?
They are different. They are different at the genetic level, they're different at the phenotypic levels. Some species like Dahlia imperialis grow 15 feet tall and make a funny looking set of dahlias that droop down at the top, dahlia flowers. So quite a bit of diversity exists across the genus. And this genus has been evolving for tens of millions of years, right? And it's moved across continents across the world. Well, Thank you all so much, and Zach and I will be around for the rest of the day.